Today is April 25th, 2017. I'm in Chicago's McCormick Place Convention Center for the annual meeting of the American Physiological Society. Today I have the honor of interviewing Dr. Randall Packer for the Society's Living History Project. Dr. Packer is a longtime member of the APS who's been affiliated with the George Washington University since 1971. He is a comparative physiologist who has studied a variety of species, and he's also taught several thousand undergraduates human physiology courses. Randall, welcome to the Living History Project.
Thank you.
And thank you for agreeing to be interviewed. So if you're ready, I'd like to ask you a few questions about your career.
Glad to do it.
Okay. So in your autobiography, you said that you were raised in Blanchard, Pennsylvania.
Yes.
And that you were the first person in your extended family to go to college. So tell me about your upbringing and how you got interested in science.
I was always interested in two things as a young boy. It was a very rural area. The town that I grew up in had 400 people. Most of my friends lived on dairy farms that were within a bicycle ride of where I lived. So I spent a lot of time on dairy farms, and the men in my family were all hunters, so we were all out killing harmless little animals and doing a sort of dissection. And the other thing that I was always interested in was how things worked. And that extended from radios to rabbits. When I was thinking about what I would do after high school, I knew I wanted to go to college, and I thought I would be a teacher. That was about as high a profession as I could imagine entering, because it was one that I had experienced. So I thought I would be a high school teacher. I did my undergraduate degree in secondary ed, but while I was doing it, I was encouraged by faculty members at Lock Haven State University to think about going on beyond the bachelor's degree. People pushed a little bit for me to go to medical school, but I came from a family of extremely limited means, and I just didn't see any way that I could pay for medical school, and I didn't even know about things like loans. But one of my ecology professors, to whom I'm very grateful, and whose name I've been trying to remember but failed to, encouraged me to go for a PhD, because he said I wouldn't have to pay, they would pay me. A little bit. And so I did. And it was a very smart decision for me, because I really do have a passion for teaching. But I'm sure I was more effective teaching at the college level than I would have been at the high school level. I was never any kind of disciplinarian. That's really how I got going. I went to graduate school actually thinking I would do ecology, because the man who pushed me in that direction was an ecologist, and I did an undergraduate research project with him in ecology. When I was looking around for a place to do my PhD, I had a nice offer from Cornell and one from Penn State for research fellowships. And the Cornell fellowship would have been studying apple maggots, and at Penn State there was a broad range of topics that I could have worked on, studying fish, reptiles, or amphibians with Bill Dunson, who turned out that's where I went, in part because of the research projects, and Bill was my mentor, and only really a few years older than I. He is a very smart man who went through undergraduate school very quickly, did his PhD very quickly, came directly to work at Penn State as a faculty member. And so when I started graduate school, I suppose I was 21, and I'm pretty sure Bill wasn't much older than maybe 26 at the most, or 25.
When you shared some things about your background with Dr. Frank, you mentioned that Bill Dunson's approach to mentoring had a profound effect on you. Can you say something about how he did that?
Yes. Bill did not try to be my friend when I was a new graduate student. He was my mentor. Although we were relatively close in age, that doesn't mean he was unfriendly. It just meant he was very professional in his dealings with me. And the very first full day I showed up to work with Bill, he sat me down and said, "Here are 3 projects. Do any of these sound interesting to you?" They all sounded interesting. I chose one working on ion balance in trout, and I started the project that day. So from the very beginning, I was working in the laboratory. And, excuse me, it was a great lab to work in. At that time, I was Bill's only graduate student, but he had a full-time lab technician. And that lab technician taught me a lot, as Bill did. I worked very closely with Bill, but he was not a hovering kind of mentor. I actually chose Penn State over Cornell because when I went to Cornell, I talked to the graduate students and they said they could never get to see their mentor. Bill was always available to me, but didn't look over my shoulder. He let me make mistakes and corrected me when I did, helped me get out of dead ends that I'd run myself into, and was just a terrific guide to how to do science and also how to have a personal life outside the laboratory. We weren't a lab that was in there from 8 in the morning till 9 at night on a regular basis. It happened sometimes, but it was a terrific experience. I was only with Bill for 3.5 years. He really pushed me through the program quickly, but in the process, I published papers on axolotls, sea snakes, and trout. And honestly, it was one of the 2 most productive phases of my career was as a graduate student with Bill and as a researcher with Mark Knepper then in the 90s, late 80s and 90s.
So what came next after you finished up at Penn State?
Went to GW, started teaching 2 or 3 courses a semester, started writing research grants, got one from NSF. I was thrilled to do it. And then I started training graduate students myself, but only master's students because I was the only physiologist in my department. There was not a large physiology department in the medical school. There were no other comparative physiologists in town. I just didn't feel like it was the right atmosphere. I've trained a couple of doctoral students, but mostly master's students who did a master's with me and then went on to medical school.
So in 1999, I understand that you took a break from GW.
I did.
Tell us about that.
Well, I just finished up being department chair for about 9 years. I guess I'd been out of the job for maybe a couple of years, and I was wondering if there was a sort of second act to my life. And I had been getting approached for jobs for a while, but I got called about a job as an associate, as dean of faculty and vice president for academic affairs at Moravian College in Bethlehem, Pennsylvania. A very nice 4-year college. I interviewed, I liked everybody a lot. The job sounded very interesting and challenging. I took it. Long story short, or actually short story short, after about 3 months I realized I'd made a terrible mistake. I was not cut out to be a vice president of academic affairs at a 4-year college. I kind of felt like, and I don't mean this as a knock on Moravian, but I felt like I had gone to the minor leagues in a way. And so I got on the telephone, frantically called GW and said, could I please have my job back? Because I had relinquished tenure, just given everything up. And they very generously said yes. I came back like nothing ever happened, had the same phone number, the same email address, everything was just as it had been. It was a very valuable experience for me. I learned a lot, and I was and shall be eternally grateful to GW for taking me back.
So please tell me about your collaboration with Mark Knepper.
I was looking to do a sabbatical in, I think, the mid-'80s, and I read a paper in, I'm pretty sure, AJP Renal, if there was an AJP Renal at that time. I don't know if that was before they split or not. Anyway, and it was about this fantastic technique developed by Mo Berg and Jack Orloff of perfusing renal tubules. I thought, wow, this really looks interesting. So I wrote Mark a letter. This was in the letter writing days. Mark wrote back, invited me out to the lab, and as Mark has done for many people in biology departments at GW and really around the world, he could not have been more welcoming. And so I did a sabbatical in Mark's lab, and then I continued to work in the lab in summers for, I don't know, 20 years or so. And it was a wonderful, wonderful relationship. It really, that's when I joined the APS and began coming to experimental biology meetings. And it really was the heart of my science career. I really did what I consider to be the bulk of my best papers came in collaboration with Mark and would not have happened without him.
And what were some of the things that you feel you contributed through that collaboration and, throughout your career? What are the things that you feel you've contributed to physiology?
Well, beginning in graduate school, I started working on the effect of acidic environments on fishes and the physiological effects. And I've continued in acid-base physiology really throughout my career. Turns out fish gills and mammalian kidneys do a lot of the same things. Gills of fish are not just about respiration. And so I have been studying ion transport and acid-base balance for my whole career. I think during my very early work, the biggest contribution perhaps was showing a tie between environmental proton concentration and sodium uptake. This was before the days of identified transporters, but I hope it helped pave the way for the discovery of the NHEs and the other transporters that are important in fish gills and in the renal nephron. In Mark's laboratory, we did some work. I was co-author on a number of papers that had to do with the renin-angiotensin-aldosterone system. We elucidated the effects of angiotensin II on sodium uptake in the proximal tubule. I think that was a modest contribution. In the world of science, I've always seen myself as a sort of minor character because I have always spent a lot of time teaching and as much time as I could on research, but probably my biggest career contribution has been teaching.
I'm so glad you brought that up because I wanted you to elaborate on that. You mentioned that when you communicated with Marty, that teaching and mentoring were important to you. So please share some of your thoughts on that.
Well, doing teaching and research as a combination just makes for a fabulous career because the rewards in teaching are immediate. If you go in there and you give a good lecture, you know it. And the students know it and they respond to it. And you get to work one-on-one with students who are starting down that confusing, fantastic path of deciding on a career. And so those interactions are just so positive. The other thing is I've always had undergraduates in my research lab, and a number of them have done really nice work. In fact, I had an undergraduate while I was with Mark, Sashank Desai, who was headed to medical school. He did a project under Mark and my supervision one summer, and during the academic year, he went to I think it was an ASN meeting, not an APS, and won best student paper in competition with graduate students. And I just couldn't have been more proud of Sashank. And he was kind of emblematic of a range of students that I've had over my career who have really gone on to, I think, terrific careers, mostly as physicians, some as physician scientists, and a few as PhDs. But most of our undergraduates are on the medical school track when they come to GW. And so pairing that sort of instant gratification with the continual frustration of doing research, it's a nice balance. The rewards of research come slowly, but when they come, they're very exciting. And I've really enjoyed a career that allowed me to do both those things.
I'd also like, if you would, for you to comment a little bit on how scientific meetings have changed over the course of your career. You said your first experimental biology, which is of course also the APS annual meeting, was in the 80s. How is that experience different from the way the meeting is now?
I'd actually like to go back to the very first meeting that I went to, which was a meeting of the American Zoology Society, as it was called at the time. It was in Bloomington, Illinois, and I drove there with other graduate students in a van 2 days to get to the meeting. I don't remember where we stayed, I think in a dorm. My talk was the first talk after lunch. And I looked out and I didn't think anybody was going to show up. And I looked out into lunch, and in the front row sat Lad Prosser, a real, sort of one of the real pillars of comparative physiology at the time. And I, of course, immediately seized up. But I managed to get through my talk. And I continued then to go to meetings. When I first switched from going to the sort of zoology meetings to coming to experimental biology meetings, I was astonished by the size. Through the years that I've been coming to these meetings, they haven't really changed in character in any negative ways. I think that they still provide the opportunity to talk to a smaller group of people who are really interested in exactly what you're interested in, or do a poster and they come by. And so you can interact with your peers in a more intimate way. And then also go to the big lectures and listen to people who do physiology in fields that I don't do, but which immediately relate to my teaching, and in which I've learned a lot. And of course, there's the camaraderie of coming to the meeting and seeing friends that you've made over the years. Most of the people at APS that I know because they came through Mark's lab.
So related to that, I was wondering if you could say something about how participation in a scientific society such as the APS has contributed to your career.
Well, I think that without a society, I just can't imagine how this would all happen. Societies have been really, I think, the bulwark of how scientists communicate, not just face-to-face at meetings, but sponsoring journals. That journal sponsorship seems to be slipping away a little bit to online places, but there are still great journals being produced by societies like the American Journal of Physiology. I hope that kind of relationship continues with societies sponsoring these meetings, sponsoring good, well-reviewed journals into the future.
One last question, which is, what advice would you give to a student starting on a science career these days?
I guess I would say the most important thing is to carefully choose a mentor by talking to current graduate students of that person, and pursue a question that you're really interested in. Because science is hard, and you really need to be driving toward a goal that you really want to achieve. So work on things that you're interested in. Although, having said that, I think anybody with the science gene gets interested in whatever she or he starts working on. And so, I think our interests change, but it's very good to have that interest and also to have a thought about what you want to do with your degree. PhDs are not just for going into academia and teaching. There are lots of avenues open for PhD students and for physiologists who learn to think kind of big picture in a way, and in a very integrative way, which I think carries over into many kinds of careers.
Well, thank you very much, Dr. Packer, and I'll let you get in a last word if there's anything else you'd like to say.
There really is not. Thank you so much for agreeing to do this interview.
Thank you.
You bet.